So it's really a great pleasure to be here today. It's my first time visiting England post-cheese in my life, so it's really exciting. And I'm really looking forward to telling everybody about the work that we're doing in my lab and hearing your feedback and hearing maybe some of the stories and sharing more in detail with you afterwards. We have been working on this project for coming up on 2 years now, and we have a small group. It's myself, Ben, and one other person in the lab, and we're coming at the question of the microbial diversity and the importance of this diversity in cheese from a different angle than Marie-Christine told you about. We're coming at it more from the perspective of seeing how we can use cheese to teach us about microbial communities in general. But I think what you'll see is that the direction we're moving is sort of in line with what Marie-Christine was doing and complements it in a number of ways. So today we'll be focusing on the work that we've started doing, which is looking at the microbial diversity and also starting to dive into interactions that are present within these cheese rinds. So we're really interested in my lab in microbial communities. And what I mean by microbial community is pretty much what you would think of for any type of community. It's a group of individuals that share an environment or some common set of interests. And the fact is that most microbes in nature don't exist in isolation. They exist as part of these communities. But the problem is that we know incredibly little about what happens within these microbial communities. So the reason that we know so little about it is because usually as microbiologists, what we do in the lab is study a species in isolation. So we take a culture from an environment, whether it's cheese or the soil or the ocean or wherever, we isolate it in the lab and we study it in great detail. Now this is in contrast to what microbes are actually doing in the environment, which is growing as part of multi-species communities. So they're not there by themselves. They're having to grow in an environment where they're surrounded by other types of microbes or macrobes or living inside of our body. So there's so many different things that can be going on, which we wouldn't necessarily learn by studying a microbe in isolation. So in the lab, studying a microbe in detail in isolation is telling us a lot about how an individual cell works. How does it gain its nutrients for survival? How does it grow? But there's a whole other set of really interesting questions that we can potentially answer by studying microbes within the context of communities. So some of the questions that we're interested in understanding for microbial communities are: how do cells interact? What's the extent and nature of these interactions? Do they communicate with each other, and how? What are the molecules that they use to do this? And how do these communities form? Can we understand some of the principles that drive the direction of different types of microbial communities? Basically, how does this all work? And the reason that we're interested, that we sort of got interested in this question of microbial communities, is because these communities are everywhere and they're important for health. So they're important for the health of every ecosystem on the planet, such as soil or the ocean. And it's increasingly aware, increasingly acknowledged, and I think it's becoming increasingly aware in the general public that microbes are important for human health. So not just in a negative sense, but also in a positive sense. So these communities that are living inside our body, on the surface of our body, are having a great impact on the maintenance of health and protection from pathogens, for example. The problem with these communities and the challenge in understanding them is that they're incredibly complicated. So in general, in a gram of soil or a gram of contents in your gut, you have hundreds to thousands of species living together, which is sort of mind-blowing if you want to try and understand what each of them are doing and how it all works. So the thing that we're trying to do with cheese is use it as a model system, and that's because model systems, just like when we study a cell in isolation, that's simplifying the equation. If we can come up with model systems to understand microbial communities, that might teach us some of the general rules about how these communities function, that we hope will apply broadly across many different types of ecosystems. So we're using cheese, and in particular we're using the rind of cheese as our model. And I'll explain why. So this is sort of where we're starting. So everybody here knows what the rind of cheese is. So this is the surface-associated community that forms during the aging process. So this is just typical 3 batches of cheese at different points of their aging. You can see the rind development over time, and the pigmentation that you're seeing is the result of the different microorganisms that are growing and colonizing the surface. This is a multi-species community. You can tell this in a number of different ways. This top picture is a scanning electron micrograph of the inside of the rind of a cloth-bound cheddar. Okay, so if you just imagine stepping inside the rind and look around you, this is what you would see. This just mess of microbes. So you have, I don't know if you can see very clearly, the light's maybe a little high, but the 2 large cells up there, those are probably fungal spores or canidia, and all the little squiggly lines are bacteria. You have different shapes, round, rods, little squiggles. Technical term for them. But 2 things to notice. One is that there are many different species there, all packed together. And the second thing is that it's an incredibly dense environment. So pretty much the rind is this densely packed multi-species community that forms during the aging process. And if you take just a small piece of the surface of a cheese and plate it out on standard laboratory medium, what you'll see is the appearance of different types of colonies growing on the petri dish, which just in a very basic way tells you that there are different species living together within this community. And so from the perspective of utilizing this as a model system, the cheese actually has pretty much everything you would want out of a model community. It's simplified in a number of ways. Compared to thousands of species, we have more on the order of tens, or potentially more or less depending on what you're looking at. We have many replicates, which as a scientist is great. There's lots of experiments. Every day a cheesemaker makes a batch of cheese. That's like 100 experiments that we can look at. It's reproducible in the sense that you're making the same type of cheese over and over again, and roughly you have a similar product, which suggests that the communities are forming reproducibly time and time again. The communities are very accessible, so we just have to go to Vermont to go get samples, or go to our local cheese shop in Cambridge. This is much easier than trying to get into somebody's digestive tract or going to the bottom of the ocean to get a sample, and much more fun. The communities can be manipulated, which I sort of think of every cheese as a manipulation of this ecosystem. And it's something that really fascinates me, that over hundreds of thousands of years of cheese making, humans have figured out all these different ways to control microbial communities, which is sort of an interesting idea. If in microbiology we're interested in controlling these communities, potentially for benefits of health or ecological systems, we might be able to learn something from thousands of years of cheese making knowledge. The communities are culturable, so I won't show you any of the data today, but it's been demonstrated a number of times in the literature that we can pretty much cultivate everything that's present on the cheeses, which is a great benefit. If you look at culture results from the soil, it's usually less than 1% of the species we can grow in the lab, so it's really difficult to study something if you can't grow it in the lab and look at it. So this represents a really powerful aspect of cheese as a model system. And another really wonderful part about cheese is that we can follow the formation of these communities over time. And Ben is going to tell you some of the data that we have based on this type of studies, but this really gives us a lot of information about what's driving the community. So I wanted to say that the more that we work with the cheese, the more we realize that the contribution here is not necessarily just as a model system for other types of communities, but that this knowledge that we're getting from these basic studies could potentially be applied in a number of ways for cheese making in general. So we think that a more detailed understanding of these types of communities can potentially help with troubleshooting problems in aging. If you know sort of the stages in community development and what can go wrong with those different stages, you might be able to alter that. It's possible that certain types of communities may be more robust, whether it's robust to changes in the environment of the aging room, or changes in salt, or even potentially to things like what Christine was talking about with robust to invasion of pathogens at different stages. And it's also possible that we might be able to identify certain species or mixtures of species that may lend interesting sensory qualities to the cheeses. Okay, so this is sort of the general outline of what we're going to talk about today. So all of this is focusing on understanding the cheese rind ecosystem. First, we're going to talk about the work we've been doing over the past year to characterize the microbial communities that are present across a broad set of cheese samples. And I'll talk about the bacterial set of data because I love the bacteria. And Ben is going to talk about the fungi because he loves the fungi. It just works out nicely. We can split it up. And then we'll tell you about the data we have on looking at the community formation over time on a specific type of cheese. And then some of what we're starting to dive into, going from the broader scale observations now to testing specific interactions that we see within the community and what we think they may be doing to determine what the community looks like. And then we'll tell you a little bit about what we see in the next couple of years in the lab. Okay, so I thought I would just give a very basic overview of the type of technique that we use to study these communities. It's fairly standard practice now in microbial ecology. We're just applying it to cheese, and we're not the only ones who are doing this, but I thought it would just be nice for all of us to know how this is done. So basically, we go and collect a sample, and our field work consists of going to cellars and going to raw and collecting samples. So we basically scrape off a portion of our community of interest, which is the rind of cheese. We put it in a test tube, and then we bring it back to the lab. When we get it to the lab, so as I told you, this rind is just a dense mass of cells. When we get to the lab, we break it open those cells by mechanical and chemical means. We release all of the DNA from those cells. So each of the cells has DNA. It gets released into the tube, and we purify the DNA away from everything else. And then we go in and we look at that DNA, and we pick up what we think of as fingerprint genes. So they're genes within the DNA that every species has that tells us what species it came from. Okay, so sort of like every cell has a little fingerprint, and then we're able to go in there and make copies of it. And then once we have copies of all of these fingerprints from all the species that were present in our community, we sequence it. And this is just a picture of the fancy sequencing machine we have in our basement. Not at my house, at our lab. So it would be kind of fun to have one now. It looks really cool. It has this like flashing light on the side, which I don't think does anything. It just makes you go, ooh, technology. It's basically the top of the line sequencing machine. It generates ridiculous amounts of data. It allows us to do the sort of large scale analysis that we're doing. And this is basically what the data looks like. This is the DNA sequence. So if you imagine the fingerprints are actually DNA sequences, and so we're able to read the DNA sequence. And then what we do is take those sequences and ask a database, who does this sequence look like? Okay, so for every single sequence, we go through and check who it belonged to. So for this particular example, this sequence, based on the database searching, belonged to Streptococcus. Okay, so probably something from the start culture. So this is the sort of process that we're doing, but on the scale of millions of pieces of DNA. Right? Yeah, a lot. We don't do this by hand. We have software that runs this analysis for us, luckily. Okay, so what we set out to do is try and perform as comprehensive a survey as we could of cheese rinds from all the major countries that are producing cheeses. So we ended up, over the course of this past year, collecting 165 different types of cheese And so for each of these 165, we tried to collect from triplicate wheels, ideally that were made from the same batch. Okay, so for each of these, the data I'm showing you is averaged across 3 wheels of each type of cheese. We tried to sample across as many different parameters as we could, so both the cheese types that we have here, the wash rind, natural rind, bloomy rind, but we also tried to take into consideration geography, so what country or region the cheese was made in, what milk type it was, so whether if it was raw or pasteurized, what animal it came from, if there were any other additives that we knew of that were used in the cheese making process. Basically as much of the cheese making technology that we could incorporate, we have that in a database that we can now try and look at. And the goal here is to try and understand how different are the communities across the cheeses, which environmental factors such as pH, salt, moisture, or geography have the most impact on what the community ends up looking like. And can we detect patterns such as co-occurrence of certain species that might suggest interactions between species that are important for the particular community? So within this 165 different samples, we ended up with 57 wash rind cheeses, 68 natural rind cheeses, and 40 bloomy rind cheeses. Okay, so this is the data. So we've been doing this over the past year, and it's just within the past month that we have finished collecting the data, and this is actually the first time that I'm showing all of the data together. And it's a bit of a mess, and I don't completely understand it yet, but there's some interesting things that are popping up, and I think over the next few months, Ben and I are going to be working incredibly hard to get this all to a point where we really understand what the patterns mean and publish it as a paper, so hopefully everybody here can see that sometime within the next year at the latest. So this is what it looks like. So what this is showing you is the composition of the bacterial portion of the community. Okay, so this is just bacteria. Ben's going to talk about the fungi. And the way this graph is representing the data is that along this axis is the percent representation of a particular species within the community. So what you see, the red is Lactococcus. So the bloomy rind cheeses have a lot of Lactococcus, and I can say why in a second, why I think in a second. But so of, say, this first sample here, Lactococcus makes up pretty much the entire community. Okay. Does that make sense? Yeah. So as you sort of decrease Lactococcus, you see other species starting to make up the community. So this is looking at the relative proportion of species within the total community. So the first set we have are the bloomy rind cheeses. In the middle are the natural rind cheeses, and this group here are the washed rind cheeses. And let's see, there's something else I was supposed to say. I'm going to dive into more detail, but one interesting thing is that so we went in classifying these based on bloomy, natural, wash, because we thought these should represent different types of cheese. They're made differently, they look different, they smell different. And it does appear to be the case that each of these types of communities has its own sort of fingerprint, right? You can kind of look and say, this looks like it's a natural rind cheese because it has this general pattern. We had a few samples that I had mislabeled as the wrong type of rind, and they stuck out like a sore thumb in our data, and I looked at it, I was like, oh yeah, that is not a natural rind cheese, it's a bloomy rind cheese. So it's sort of interesting that there are these very general patterns across the different types of rind. And these are, and then what I've done here, these are the, I think 14 most abundant species that we find in the cheese, and these are the things that are above 1%. 1% or greater on average across all the samples. So there are other things present in various cheeses, but if they weren't on average above 1%, I don't have them included in this data set. but I have all the data for, there are more species. This is just across all of our samples, this is the general most abundant bacteria. So this is just a very basic calculation of the average diversity across the samples. So this is calculating across all the samples in the first bar. So across all the 165 samples, on average we have about 8 species, although there's probably more than that. So the type of sequencing that we're doing doesn't necessarily give us enough resolution to say species. So my guess is that within some of our groups there are several species. So the numbers are probably a little bit higher than this. And then I just split up the different rind types based on raw versus pasteurized, because I thought that might be something people would be interested in seeing. So within the bloomy rind samples, we had 15 raw milk cheeses. Their average diversity was around 7. And we had 25 pasteurized cheeses, and their average diversity was 4.5. So a little bit lower diversity in the pasteurized milk cheeses. In the natural rind cheeses, it looks about the same, whether the milk is raw or pasteurized. So that might say something about where the microbes are coming from. It might be the case that for natural rind cheeses, maybe they're coming more from the environment other than the milk, but we're not really sure what this means. And then for washed rind cheeses, you actually have pretty big difference. For raw milk washed rind cheeses, we have on average 10 species, and for pasteurized, we have on average a little over 6. Okay, so I thought I'd just walk you guys through a few of the very first interesting things from the data set that we're finding. So a few microbial surprises. It's always nice. After lunch, a few microbial surprises. So one surprise is the lack of starter or adjunct cultures that we're seeing. So what basically this is saying that most of the microbes that we're seeing in the rind are coming in from the environment, whether it's the milk or the aging room or the cheese house. So I've highlighted in yellow Lactococcus and Streptococcus, which we're fairly sure these are coming in as starter cultures, and pretty much we only see them in the bloomy rind cheeses. So we are just trying to sample the rind in the study, so we shouldn't actually see lactic acid bacteria, but I think what's happening is that a lot of bloomies have very soft rind, so we're sort of inadvertently getting curd in our samples. And also, we think that the bacterial diversity, the community on the bloomy rind cheeses is really dominated by the filamentous fungi. So there really aren't that many bacteria in the rind. So I think all the signal we're getting is just from the sort of errors in sampling the curd. But across the rest of the cheeses, we really don't see very many starter cultures added to the cheese. So a lot of indigenous microbiology appearing on the surface of the cheeses. Okay, next surprise is Brevibacterium. So I think we always think Brevibacterium is on washed rind cheeses, right? That's the stinky red thing that we think of as being associated with washed rind cheeses. But actually, it's really important in the natural rind community. So this is sort of a surprise to us that it's such a dominant number of natural rind cheeses, and it's actually not that abundant in washed rind cheeses. So yeah, which is it's surprising, right? And I'll tell you what is abundant there. But yeah, so it's in a few bloomy rind cheeses. It's very, very important in the natural rind cheeses. And we'll go into more detail about that actually later. And then it's a minor portion of the community in washed rind cheeses. Okay, so the next surprise is that we have a lot of staph in cheese. And this is not Staph aureus. This is coagulase negative staph species, which are not toxin producers. They're staph that are present in the environment in many different places. And we sort of feel like they're underappreciated in terms of their importance in the cheese production and aging. And they co-dominate with the brevibacteria in the natural rind cheeses. They're very, very frequently found in natural rind cheeses and fairly abundant. We see them in some wash and bloomy rind cheeses as well. Okay, the next surprise is that the wash rind cheeses have tons of ocean bacteria. And so I think this goes back to what Marie-Christine was saying, is that possibly the salt is a major source for inoculation onto the cheeses. So what I've highlighted here are these 4 different types of bacteria, which if you look in the databases, their closest relatives are found in the ocean. Okay, so Sphingobacterium, Halomonas, Pseudoalteromonas, and Vibrio. These are all ocean bacteria. So Halomonas is from the Arctic Ocean. That's the closest relative to the cheese-making strains that you see. So we have this group of ocean bacteria which are really, really frequently found in these cheeses. We don't know what they're doing. We don't really know what they're contributing to the cheese or to the community, really. So it's an interesting kind of observation. Okay, and then I thought everybody would want to know, what about pathogens, right? Can we find any pathogens in our samples? And actually, we didn't find anything. So it's sort of a relief to me, but we didn't see Listeria in any of our sampling, and we didn't detect any Staph aureus. So I don't know if it's because we're mostly dealing with aged cheeses, so these are cheeses at the point of sale. So they've already been aged out for however long they're going to be, so that probably also decreases any incidence of pathogens, but this is, I think, really interesting. Across 165 times 3 samples that we took, we didn't detect any pathogens. And then this is a sort of another way that we're starting to analyze data, that we're starting to look at the patterns of species within the community. So what this is it's a tree, sort of like a family tree of these communities. Okay, so basically 2 communities that look very similar will be grouped together. And then on the bottom of this, I've just listed the sort of a heat map of the abundance of these different species across these clusters. And we're seeing groupings by rind type in a lot of cases. So there we have a natural rind grouping here, which seems to be sort of clustered based on the co-occurrence of staph and brevibacterium. We have this really interesting and unique community with washed rind blooming cheeses. So something like the Langre, which I'm not pronouncing right, or a red hawk. So these cheeses that are inoculated with filamentous fungi but then washed at some point during the aging period have a totally distinctive community on them. And they are really dominated by these ocean bacteria. So they have Halomonas, Microbial Diversity, Vibrio, and we think that it's not just the washing that's doing this, because there's other cheeses that are washed but that don't really develop this distinctive community. We think what it is that it's being driven by the presence of filamentous fungi. And the reason that we think this is sort of an interesting observation is because for several of these bacteria, especially Vibrio, they're known to be in the marine environment and utilize chitin. So they're known to grow on the surfaces of crustaceans. So Vibrio just loves to grow on shrimp shells out in the wild, in the ocean. And you might think that maybe shrimp and cheese don't have a whole lot in common, but filamentous fungi that are growing on the surface of cheese, their cell walls are made of chitin. So we think that the presence of the filamentous fungi might stimulate the growth of these particular types of bacteria, because the bacteria are able to grow and use the chitin potentially as a food source. So that's a hypothesis that we're testing in the lab right now. Okay, we have a bloomy rind cluster, which is basically just lactic acid bacteria, and then we have this mess of things that we need to figure out what it means, but a combination of washed, alpine style, and natural rind cheeses that have the more complicated community type. Okay, so that's the bacterial side of the story, and then Ben is going to tell you the rest of what we have for now. We can both take questions at the end.
So even though I like to think of the filamentous fungi and the yeasts as sort of the majestic oaks of the cheese rinds, they tend not to get as much attention as they should, because mycologists like myself are actually quite rare. But fortunately, Rachel was crazy enough to hire a mycologist to try to figure this out. And unfortunately, because fungi are not as well studied as bacteria, we don't have as many tools to study them, and so we've actually been doing a lot of methods development. So we've been using the same approaches that Rachel described, but we had to go back and sort of reinvent the wheel because it's the fungi and they don't get as much love as the bacteria. So I won't be able to tell you as comprehensive a story about the bacteria today because there's a machine in our basement back in Cambridge cranking through millions of DNA reads right now as we speak of the fungi. But I can give you some snapshots of what we know so far. So what I'm going to do is show you another colorful graph, just like Rachel was showing you. And here is the data for the fungi. So again, we're looking across the bloomy, natural, and washed rinds. And this is showing you lots of different colors representing the different genera or species of the fungi. And what you'll notice right away is something very similar to what Rachel presented, is that different types of rinds have very distinct signatures for the composition of the fungi. So the bloomy rinds tend to be dominated by the blue thing, which is not surprisingly Geotrichum or Geotrichum, depending on which side of the Atlantic you're from. The natural rinds tend to be dominated by Scopulariopsis in yellow, which is that thing that makes those beautiful spots if you're a mycologist, or those dreadful spots if you're a cheesemonger across the rind. And then on the wash rind cheeses, we have a crazy mix that we're trying to sort out. So I'm going to break this apart for you and explain a little bit more in detail, get rid of some of the color, and show you the patterns. And I just wanted to point out that these are our most dominant genera. So not surprisingly, Dibariomyces, DH, which is added as a starter culture, Geotrichum or Geotrichum, Scopulariopsis, Fusarium, which is a really interesting story I'll tell you a little bit about, and of course Penicillium, the mold we all love. So this is showing you the distribution of just Geotrichum in yellow, and in black are all the other fungi. And as I said already, Geotrichum is very dominant on the bloomy rind cheeses. It's often added as a starter culture. And we also do see it in washed rind cheeses where it's also often added as a starter culture. And what was interesting to me is that in the natural rind cheeses where we generally don't add Geotrichum as a starter, we do sometimes see interesting blips of Geotrichum, especially on a couple of cheeses that we sampled. These are very rustic Italian cheeses. And what's especially interesting to us to follow up on some of the work that Sister Noella Marcelino did on Geotrichum is that we're finding some really interesting endemic wild strains of Geotrichum on these natural rinds. And these could potentially be interesting strains to use in the future as starter cultures. These are things that have not been spread around the world by Danisco or other companies. These are things that are probably coming in from the environment on leaves or from the soil. So these could be really interesting for generating new and interesting flavors. So we're trying to explore that a bit more. Looking now at just Penicillium, we do see Penicillium in the bloomy rind cheeses, not as abundant as Geotrichum. Part of that could be our methods. Penicillium actually has a lot less DNA per cell than Geotrichum, so we may pick up more Geotrichum simply because of the amount of DNA it has. We also see various species of Penicillium on the natural rinds, not surprisingly. Some of those are blue cheeses, so we get Penicillium roqueforti. And we see a few blips, but very rarely do we see Penicillium on the washed rinds, which makes a lot of sense. Not surprisingly, Camemberti is the most abundant on the bloomy rinds, and we see a bunch of different Penicillium species on the natural rinds. Now to Scopulariopsis, again, that beautiful to me, but not so beautiful to some, mottled brown mold that we see is very common on natural rind cheeses, especially in the late stages of succession as the cheese is developing. And this is the mold that often can grow in large abundance and start to penetrate into the curd and cause a lot of serious problems. But we find several different species of it, and it's very common on the natural rind cheeses. We see a few blips on the bloomy rinds, and we also do see Scopulariopsis on the washed rinds. And here, this is what I am really fascinated by right now and trying to explore a bit more, is this genus of fungi, the Fusarium. So Fusarium is something that we commonly think of as growing out in soils, and I didn't really think about Fusarium growing in cheese before I entered this project. But what we were finding in the lab is we'd go to a washed rind cheese like Windermere from Jasper Hill, and we'd grow it up. And most of the things on a washed rind cheese are not filamentous. They don't grow like a mold because it's being washed, and it's a bad place to be a mold if you're continuously being washed. We find yeasts and bacteria. But we ended up finding this thing called Fusarium domesticum. And this is a mold that actually burrows into the agar when we grow it in the lab. And so what we think it's doing is it probably grows sort of below the rind. It's actually kind of being protected from the washing. And some research in Switzerland and a little bit in France has suggested that it has a great effect of reducing smeariness and stickiness of the cheeses. So sometimes when a cheese rind gets really sticky as you're trying to manipulate it, move it, and flip it, you get these really sticky rinds that are problems for rind stability. And when you add Fusarium, it actually gets rid of that problem pretty consistently. So we're really excited to learn more about the biology of Fusarium and its role in helping cheeses and cheese rinds be stable. So I'm really excited. I talk about Fusarium all the time now. It's like my new favorite fungus. Yeah. And finally, of course, we have the yeast. So these are the single-celled, not the fuzzies, but the little balls of fungi. And not surprisingly, they're abundant on washed rinds. They are not very abundant on the bloomy rinds, and that's because a lot of Penicillia and Geotrichum fight off the yeast. They actually inhibit the growth of the yeast, so we tend to not see very much yeast in the bloomy rinds. But we find a lot of them in the natural rinds. We find both Deveromyces, Cleveromyces, and some Candida species that are there. And the most common yeast that we found was Deveromyces. Not always added. Sometimes it's a starter culture, but sometimes I think it's coming in from the environment. So the data that both myself for the fungi have presented and Rachel presented are from this stage of the cheese, the final time point, the delicious time point that is the end of aging. But of course, as you know, if you make cheese, that's not what it always looks like. Very early on, such as with the case with Bailey Hazen Blue, which is a cheese we've worked a lot with, there is no rind. It's a naked cheese to the eye of Rachel and I. And slowly over time, you get to development of a crust, as we see in Bailey. And eventually that crust starts to age even more, and you get to this beautiful stage of fuzziness, which I love, where the rind is really flourishing with filamentous fungi. And then eventually you get the Scopulariopsis coming on, the mottling that we see here. And then maybe it's brushed, and this is the final cheese. So there are dynamics, there are microbes that are coming in and out during those stages of aging. So by just sampling the end time point, we're really sort of missing some of those dynamics. And so what Rachel and I wanted to do was to work across that succession of microbes, the succession, the change over time of the microbes, and understand how that was changing over time. And we focused on Bailey Hazen Blue because it's a cheese that we have access to. We have great budding microbiologists up at Jasper Hill that were able to do the sampling for us. So Matteo and his crew actually went in and took the samples for us because Rachel and I have to get stuff done back at the lab. And so what we have now is a portrait of the microbial composition over time. So what I'm showing you here are the different types of bacteria that we see on Bailey Hazen. This is just for the bacteria now. And I'm showing you across the weeks of aging. So this is over a 10-week period. This is not sort of the first week where it's setting in the cheese house. This is after it's been in the cheese house and moved over to the cellars. And what we see is initially we mostly have Lactococcus in the very beginning, the starter culture. There's no rind there. Really, we're just sampling the curd. But in weeks 2 and 3, we start to get a proliferation of these Staphylococcus, these underappreciated bacteria that are on the rind. And what we're learning in the lab is they're actually playing a huge role in deacidification of the rind. So we're finding that they can deacidify as much or even better than some yeasts. So they're probably doing very important things. And then by weeks 5, 6, and 7, we start to see the flourishing of these Brevibacterium on the natural rinds. And by the end of aging, by week 10, we actually have about equal mix of Staph and Brevibacterium. So again, as you can see, if we just look at week 10, what we see at week 10 is a very different picture of what we see at week 3. And of course, what we can do is then add the fungi in and make comparisons between the succession of the bacteria and the fungi. So for the fungi on Bailey Hazen, what we see is in week 1, very, very huge dominance of Candida catenulata. And what's really interesting is that in Bailey Hazen, Clavieromyces lactis is added as the yeast that's supposed to be the major rind yeast to deacidify. Turns out K. lactis is not able to tolerate these wild yeast of Vermont. In Canada, Cateniolata turns out to be what we call a killer yeast. It actually produces toxins that kill off other yeasts. And when we grow it in the lab, K. lactis is just not able to grow at all. It kills off K. lactis. And so initially, for the first several weeks, we have Candida cateniolata growing with the Staphylococcus. And then we see a little blip of K. lactis right there. It tried to come out, but not much happened with it. And eventually, we start to see the fuzzies coming in, the penicillium, and then that beautiful Scapulariopsis coming in towards the end of the ripening. So again, we have a very different portrait from the very beginning of the cheese and the very end of the cheese. And what Rachel and I have been doing is starting to look at these patterns and ask questions about, well, are these fungi and bacteria potentially interacting over time? And how are those interactions shaping the development of the microbial communities? So we sort of have this conceptual view of that succession. Again, the lactic acid bacteria, our yeast, Candida, and a little bit of Deberomyces we see blips of. And then the Staphylococcus, which we're putting right in the center because we're spending a lot of time focusing on them. And then our filamentous fungi, which increase over time in abundance. And then we have Brevibacterium and a little bit of Crinibacterium, these actinobacteria, as we call them, that increase over time. And what we've been doing is focusing specifically on Staphylococcus. So if you zoom into a wheel of Bailey Hazen blue, where we have this crusty stage, and this is something we actually see in a lot of cheeses. So if you have this early crusty stage, it probably is composed a similar composition to what I'm going to talk about. When we zoom in and look at that under the microscope, we have large yeast cells. This is the Candida cateniolata. And all these little dark dots here are the Staphylococcus species. And these staph are beautiful. To a microbiologist, these are stunning. It's not just one staph species. It's 4 different really beautiful staph species. Staph saprophyticus, which is commonly found in the UK on cheese rinds. Staphycorum, which we heard about earlier today. Staph xylosis, and Staph succinus. And none of these are added as starter cultures. These are all coming in most likely from the milk. We don't exactly know yet, but they're coming in from somewhere. And we find these different species co-occurring, growing together on many different cheeses. So it's not just a phenomenon we're seeing on Bailey. We're seeing this on many different cheeses that we're sampling. When we look at the abundance of these different species, we find that Staphycorum dominates. For some reason, even though there are 4 species growing together, Staphycorum is the most abundant. So 74% of the Staphylococci are Staphycorum. So what Rachel and I are trying to do is understand why. So we're asking questions about why these different species show up in different ways on the cheese. So first of all, how are these 4 different species coexisting? Why are there 4 Staphylococcus? And then why is one species dominating over another? And what we're trying to do is develop a model. So we talked a little bit about predicting microbial communities earlier. We have a conceptual model that we're trying to use to understand the interactions between these different microbes with a focus on these staph. And so we're thinking first about migration. How are these microbes getting to the environment? Is it through the milk? Is it through starter cultures? We know for the staph the answer is no. Is it from the environment? And then we can add on to that equation, we can think about the way that these different microbes tolerate the environment. As we change the salt, as we change the moisture, how are those influencing the interactions between these different species? We're also thinking about microbial access to resources. So microbes are initially really limited by iron when they're growing on a cheese rind. There's very low iron availability, so we're doing some work in the lab to address that. And what we're mostly looking at are interactions between species. So how are these different microbes detecting each other? How are they communicating with each other? And sometimes how are they fighting with each other? And ultimately, we'd like to be able to put some math behind this and actually make predictions about how microbial communities develop based on this very simple model. So very quick vignettes that we're seeing from the interactions only. We're not going to talk about anything else. We're seeing some interesting interactions between the staph and the lactic acid bacteria. So if we look across the 4 different staph species, what we see is that Staphycorum is very strongly inhibited by lactic acid bacteria, which is sort of counterintuitive, right? It's the most abundant staph, but it seems to be inhibited early on. So this is showing you when we grow, these are lawns of staph, and what we've done is we put a spot of the lactic acid bacteria on that lawn of staph. And so you can see this little area here where there's no growth is because the lactic acid bacterium is inhibiting the growth of the staph. We're also finding that Staphylococcus has somehow an arsenal that it uses against the other staph species. So this may explain why it becomes so abundant. So when we do the same kinds of experiments, but when we put Staphylococcus growing with the other staph species, it creates these same zones of inhibition where there's no growth of the staph. And we think that Staphylococcus has some antibiotic compound that it can produce, maybe the same that's used against Listeria to inhibit the growth of the staph species. I think one of the most exciting and the strongest signals we've seen so far are from this lovely Scapulariopsis in the staph. So what we've done here, an intern in our lab this past summer grew up different filamentous fungi. So he grew up Geotricum, Penicillium roqueforti, Penicillium commune, and Scapulariopsis. These are all fungi we find on Bailey. And he took little plugs of those and placed them on top of the staph species growing on petri dishes in the lab. And what you can see is there are these massive zones of inhibition. These are places where the staph is unable to grow. This is like very striking in microbiology world. And what's really interesting is Staphylococcus is one of the least inhibited staph species. So it seems to be able to tolerate whatever antibiotic that Scapulariopsis produces. So if you think back to that model of succession that we showed, where the filamentous fungi come in and the Staphylococcus start to go away, perhaps Staphylococcus is able to tolerate that inhibition at the very end of the aging, and that's why we see it being most abundant. So these are just some very preliminary interaction studies that we're doing. Of course, we want to understand the whole community. These are just pairing different species together. And so what we're doing in the lab, and what Julie, who's the other member of Team Microbe, is doing back in Cambridge right now, is actually pulling all the species together and putting them in an entire community. So not just staph and Scapulariopsis or staph and lactic acid bacteria, but all these species. And we're developing model communities in the lab to understand the interactions that are driving the development of Bailey. So what she's been able to do is take this pattern of succession, the transition from the blue there, which is Staphylococcus, to Brevibacterium, which is red, and she's actually been able to recreate that succession in the lab. So she's done it in petri dishes like these, where she gets the same exact transition from staph domination to Brevibacterium domination. And what we're going to try to do is use some of the genomic techniques that we have in the lab and other methods to tease apart why these things are happening and layer on more complexity by adding in the filamentous fungi, adding in the yeast, and trying to get more like cheese. And eventually, hopefully, doing experiments on intact cheeses, perhaps at Jasper Hill or other places. So just to sort of summarize across what both Rachel and I talked about today, we're doing this large-scale comparative study of the rinds, and we're starting to see some interesting patterns and developing a lot of hypotheses. As Rachel said, we find more things and we have a lot more questions, so we're just continuously overwhelmed with exciting new questions, which is great. Most bacteria in the cheese, as Rachel talked about, are not inoculated, so we have this huge influence of bacteria coming in from the milk or from the environment on the cheeses. One of the microbial surprises that Rachel talked about is this dominance of Brevibacterium on natural rinds. So that might change our sort of thinking about this orange-red bacterium that's out there and playing a role in rinds. And we see that the method of aging, the different types of rinds, and maybe also raw versus pasteurized, has a significant impact on the type of community that we see. We're also starting to see some fungal signatures for the different rind types, and as we get the complete data set, hopefully we'll continue to be able to explore that. And we're also going to start looking for co-occurrence patterns, not just between the different bacteria, but between the bacteria and the fungi to tease apart more interactions. And we're now doing this sort of hypothesis testing in the lab to understand this pattern of succession that we see, to look more at these bacterial-bacterial and bacterial-fungal interactions. And hopefully we'll be able to use that equation to actually predict microbial communities. So if we say we're adding these 10 species together, we should be able to get this microbial community. And hopefully that kind of understanding will help the cheesemakers better manage rinds. So we'll be able to understand defects, we'll be able to understand why certain rinds have certain aromas. And so we're hoping that all this basic science that we're doing will be able to directly feed into the world of applied science. So with that, we'd like to thank you, and we'd also like to thank people that have helped out with this project. Julie Button, who's an excellent scientist, but also a great milker. And Marcella, who is a PhD student from the University of Parma that came to help us out. She did a lot of the work with the Bailey succession that I talked about. And Jasper Hill, which has been instrumental for allowing us to come up and take samples and listen to us and brainstorm with us about microbes and cheese. And Rachel has been generously funded by the FAS Center for Systems Biology to start her lab, and then to hire mycologists, which has been wonderful. And we especially like to thank all the cheesemakers that have sent us samples and helped us be able to get this broad survey of cheese. And we're also looking for, if you have any other questions or you see any patterns that might fit into some of the stories we talked about today, we'd love to hear more from you about that kind of stuff. So thank you.